Previously, we have talked of the unit membrane model proposed by Robertson, and before that, we have also talked of the sandwich model proposed by Danieli and Dawson. Both the models were rejected, and now the foolproof model, that is what we call it as a fluid mosaic model, has came into existence. I already told you that more than 50 models has been proposed regarding the structure of the plasma membrane, but 3 models are very important. In NCERT, only fluid mosaic model is given, but the 2 models, that is sandwich model and the unit membrane model, was very important. So that's why we have discussed. So fluid mosaic model was proposed by Singer and Nicholson in the year 1972. So this was proposed by Singer and Nicholson in 1972. What they did is they used the same information. Of Danieli and Dawson, that is basically the sandwich model. Using the same information of the sandwich model, but they have interpreted it in a different manner, which Danieli and Dawson has not thought before in that particular time when they have discovered the model. So basically, Singer and Nicholson have used the data of Danieli and Dawson, but interpretation was different. What they said is that plasma membrane is made up of 2 things same, proteins and phospholipids. Proteins are alpha globular proteins, the same bonds, that is proteins are of alpha globular type. Now, what additional thing that they have given regarding the protein is they have divided proteins based on the location, that whether the protein is located interiorly, whether the protein is located exteriorly. So, on the basis of the location, they have divided proteins into 2: intrinsic protein and extrinsic protein. Intrinsic protein, that means which is located within the membrane, either partially or completely. So they are embedded partially or fully in the phospholipid bilayer. In the bilayer I am writing, that is within the membrane. And extrinsic is located on the external side, on the exterior side of the plasma membrane. That is basically they are attached to the head region, right? So they are located on the outermost side, attached to phospholipids head. Attached to phospholipid head region. Attachment is again a loose kind of attachment. The third information, that is again PL, phospholipids are amphipathic. So phospholipids are amphipathic. That is, they have hydrophilic and as well as hydrophobic and present. Right? Now again the structure, that the head region, phosphate is attached, and this is the tail region. Hydrophilic, hydrophobic and the phosphate group is attached. That together make the phospholipid bilayers. Right? Now regarding the phospholipid, additional thing that they have given is movement of the phospholipid was explained by Singer and Nicholson in the fluid mosaic model. So they have also given an information regarding the movement of phospholipids, and based on this only the dynamic nature of the membrane, which was not explained by any of the model, that is either sandwich model or unit membrane model. If the phospholipid proteins are moving, then and then only the membrane would be dynamic, and that is what we want, because the membrane is a static solid membrane, no transportation, no movement is at all possible, right? So the PL shows two types of movement, that is phospholipid shows two types of movement, that is transition movement and flip-flop movement. Transition movement and flip-flop movement. What is basically transition movement? That is movement of phospholipid molecule in the same layer. So let us say if few layer I am drawing, if the phospholipids are moving within the same layer, that is, say for example, if this is one phospholipid, this is moving here and this is moving here, then that arrangement or that movement is called as transition movement. That is transition movement, that is movement of PL phospholipid in same layer. Flip-flop movement is basically the movement of phospholipids in the two layers. That is, if this is moving here, then this will move here. So flip-flop movement is movement of phospholipid in two layers, that is bilipid layer. Arrangement of phospholipids. Again, force of attraction between these PL phospholipid is of weak van der Waals force. So weak van der Waals force is there. Attraction between bilipid layer is there, right? Then they have additionally the pore size. Arrangement is the membrane, how it is arranged: protein, phospholipid bilayer, then again protein is present, right? Protein, phospholipid bilayer and protein. If you see, alpha globular protein are of two types: intrinsic proteins as well as extrinsic proteins. This is very important. Intrinsic protein are embedded within, that is partially or fully they are embedded within the bilipid layer. Extrinsic are on the outermost side layer only. That is, they are basically attached to phospholipid head region. That is this region. The proteins are attached as this. If it is of alpha globular type, the globular proteins will be attached. Now, extrinsic, intrinsic can be given as peripheral or integral proteins also. So, intrinsic, or you can also write it as integral protein, because it is integrated within the membrane, either partially integrated or fully integrated. That also we will draw in the diagram. Extrinsic will be peripheral protein, because they are located on the outermost side, on the external periphery it is located. So, integral as well as peripheral proteins. This intrinsic and extrinsic was on the basis of the location. Integral and peripheral is based on the ease of extraction, right? Now, if we have to extract integral and peripheral protein, so which protein will be extracted easier? Of course, peripheral protein will be extracted easier, because this protein is located exteriorly, that is on the periphery. Let us say here. So simply you can use the detergent to extract this protein. Then integral protein is fully embedded either, or it is either partly embedded within the membrane. So in order to extract integral membrane protein, you have to break the membrane first, and then you have to extract the protein from that membrane. While in case of this peripheral protein, only simple using the detergent we can extract the proteins that is located on the external side of peripheral proteins. Right. Now let us see the checkpoints, whether this model has to be accepted or not. For that we have to draw the model first. These are the head region. Tails are facing towards each other. All these are tails. Proteins are of two types, that is intrinsic as well as extrinsic. Extrinsic proteins are basically the alpha globular type proteins only, which are more in number. So on the both the sides these proteins are located. And what about this integral protein? They are either partially or fully embedded. So we will draw one partially embedded protein, and let us say this protein is fully embedded protein. Right? We will label also. So this is basically an integral protein, or you can say intrinsic protein. This is extrinsic protein. This is what we call it as the bilipid layer. Right? Now we have to check the first point: entry of fat and water soluble molecule. Fat soluble molecule can easily enter. Both the models have shown, whether it is a unit membrane model or sandwich model. Fat soluble substance are not creating. So that means membrane is not creating any problem for getting in or letting in the fat soluble substance. Now what about water soluble substances? Now water soluble substance can easily enter through these proteins. Right? So both fat and water soluble substances can easily enter the cell through the fluid mosaic model, that is this model of plasma membrane, right? And these proteins are basically acting as a channel, a passage through which water soluble substance can easily enter the cell and it can come in. So they are also called as channel proteins. Who are called as channel proteins? Basically these intrinsic proteins are also called as channel or channel proteins. Then P to PL ratio we have to see. P to PL ratio. For considering or discussing the P to PL ratio, let us say we will consider only this region. How many proteins? 1, 2, 3, 4, 5, 6, 7. 7 proteins are there. Corresponding phospholipids are only 4. 1, 2, 3, 4. Not 7, because this protein also we have to count. So it is basically 8 protein to 4 phospholipid ratio is there. Proteins can also move, right? Because it is acting as a channel. Phospholipid bilipid layer, it is also moving. Transition movement, flip-flop movement. So membrane is not static. So second and third point are fully satisfied. That means this model has to be accepted. So second point was P to PL ratio is a change ratio, and any molecule can move. Straightforward, we can write the third point also based on the second point discussion, that if the P to PL ratio is variable, although it was variable there also in both the models, but they could not explain the movement. And Singer and Nicholson has given the movement, that is flip-flop movement, transition movements, extrinsic as well as intrinsic proteins. So membrane is dynamic. Dynamic means that is membrane is not a static solid membrane, it is a dynamic membrane, that is it moves. And that's why this dynamicity, the moving property is because of the fluid that is present. That's why it is called as fluid membrane model. Right? Now dynamic fluid nature. What Singer and Nicholson used a phrase in their model, that is protein icebergs in the sea of lipids. Phrase that they have used is protein icebergs in the sea of lipids. So this lipid bilayer they are resembling to a sea, and these proteins are basically resembling the icebergs, and they are moving. How they are moving? Because of the fluid nature of lipids. And this word, there is one more word that is given is quasi fluid nature of PM. Quasi fluid nature, that is basically semi-solid nature of the plasma. That is membrane is neither completely solid, membrane is neither completely liquid. Membrane is basically semi-solid, and the word that was used was quasi fluid nature that is associated with the plasma membrane is transportation of molecules. So transport across plasma membrane. Transport across plasma membrane is basically divided into two, that is passive transport and active transport. Passive transport and active transport. Passive transport, as the name suggests, it is a passive movement without expenditure of energy. So energy is not involved. Energy in the form of ATP, that is adenosine triphosphate, is not involved. Here in active transport, energy in the form of ATP is involved. Right? If energy is not involved, the movement would be slow. So it is basically a slow movement process, and this shows fast movement. Energy is not involved. Concentration, that means movement, but transportation is across the concentration gradient, that is with the concentration gradient. So transportation or movement is towards concentration gradient, that is basically from high to low concentration. Here movement is, since energy in the form of ATP is required but it is utilized, so basically from low concentration to high concentration it can go. So movement is against the concentration gradient. Here it is towards the concentration gradient. Movement is against the concentration gradient, that is low concentration to high concentration. Here semi-permeable membrane may be present, may not be present. Here semi-permeable membrane is must. Semi-permeable membrane may not be present. Presence of semi-permeable membrane is compulsory. So presence of PM, that is plasma membrane, is mandatory. Right. Now, this is not affected by other external factors like temperature, pressure, absence of the O2, certain metabolic inhibitors like cyanides. So, not affected by factors like temperature, then absence of O2, inhibitors like cyanides, which is metabolic inhibitor, but this is affected by all these things. Right? Now, this passive transport is again of three types. Passive transport is again subdivided into three. That is the normal diffusion of solid, liquids and gases. If semi-permeable membrane is involved, right, and the movement is water as a molecule is moving or any fluid, basically water only, then we call it as osmosis. And the third one is facilitated diffusion. What is the difference between facilitated diffusion and the normal diffusion? The concentration gradient is there, that is from high concentration to the low concentration. But in case of facilitated diffusion, some facility is provided, as the name suggests, and this facility is provided by the transport or the channel proteins. While in case of normal diffusion is just along the concentration gradient. Active transport, you can take many examples like sodium potassium pump, glucose, which is the high energetic rich molecule in the form of ATP. One glucose molecule provides approximately 36 ATPs in the cell. So that glucose is transported by what? It is a water soluble substance. To is transported through sodium potassium pump only, since membrane is bilipid layer. Water molecule cannot be easily transported, but we have seen that intrinsic proteins are acting as a protein channels. So sodium potassium pump is responsible for transport of glucose. Alone pump are also example of active transport, like sodium pump, potassium pump, then you can use calcium pump. Exo and endocytosis are also example of active transport. Exo and endocytosis. Exo means something which is going out of the cell. Endocytosis, that something is coming within the cell, right? And later on, one more thing I have to add here is regarding the RBC cell membrane composition of the proteins as well as lipids. When this model was proposed, they have also studied, they have estimated the chemical analysis of the RBC membrane, that is erythrocytes. Chemical analysis of the erythrocytes, or it is also called as RBCs. It is made up of 52% proteins and 40% it is lipids. Apart from the proteins and lipids, it was also discovered that membrane also contains certain saccharide, that is carbohydrate molecules. So this was all about the structure, function